Afternoon and welcome to everybody for joining us today. This is, as you know, the first time that we are trying out one of these phylogenetic sessions. The idea is that if it's helpful, that we will have similar sessions and that we go through the various processes. So today we're going to start at the very basics of how to read the phylogenetic tree, what kind of terminology to use, but afterwards we plan, if you're interested, to go on to things like in more detail how to construct a data set, what kind of approaches you can use to construct these trees, what's bootstrapped, what's the meaning of it. We already had a number of those questions, and I think a bootstrap session will probably be a very popular one. But great work of thanks to Martin Coetzee, Professor Martin Coetzee, for being willing to help us start this process, and I hope that you will enjoy it. The lecture itself will be very short, and then we open for questions. So we will continue as long as you have questions, and we might have answers. So, but I trust Martin will have. So, thank you very much, and over to you, Martin.
Thank you, Flonis. Yeah, so also a warm word of welcome from my side on behalf of the SASSB. And especially to, well, actually to everyone. But I want to say really thank you to all the students that are participating, and some of them are sitting here in the room, so thank you guys, or people, I say guys. So this session, as you've heard from Flonis, is about reading phylogenetic trees and the terminology that we use in describing relationships and our interpretation of these trees. And I thought that would be a good starting point, because even if you do not do phylogenetic analysis, most of you are reading papers with phylogenetic trees in it. And I think it is necessary to understand how to interpret those phylogenies and their relationships. So that will be our starting point. It's an informal discussion. It's not a lecture per se or a seminar. So you're welcome to interrupt me or just put up your hand if you want to ask a question. Something else that's important is that you should please let us know if you cannot hear me due to our sound system, or perhaps I'm getting a bit tired and then my voice goes down. So you're welcome just to indicate if you cannot hear us or me. So let's start. So reading phylogenetic trees and the tree terminology that we use, it is an overview. If I could get my slides to work, it will help. So some of us here in the audience are also second year students or third year students, and they do not know what a phylogenetic tree is. So I thought it should be important to just define it. So it is a representation of the genealogical relationships among species, even among populations or individuals. And these trees can be presented in different ways. Here I have a rectangular tree that most of you are familiar with. These circular trees that are often seen in publications where we have huge data sets, or large, not so much a data set, but a large number of taxa. And if time permits, I will show you one of these trees that was published in Science. So it makes it very easy then to use less space, but also to compress many of these taxa in a visually understandable way. And then in the past, in the older literature, you often see these triangular trees. They all say the same thing. They all say this presents the same relationships, A and B being close related, C and D close related, A and B sister group to C and D. And if you look at these trees, They are all the same. So it is important to understand what nodes and terminal nodes are and branches and in-groups and out-groups and what's the root. Here is a phylogenetic tree. It's a very simple one, just to explain these terms, words that we use. So the blue nodes are internal nodes. They are the common ancestor before divergence took place. Then we have the terminal nodes. That's the present day organisms or fossils even, if you have a record or characters for that fossils. The branches are connecting the taxa. And as I've said, the nodes are then the ancestor of the taxa going down the phylogeny or down the lineage. So in this case, this node with the star is the ancestor or the hypothetical ancestor of taxa A and B. This black star is then the ancestor of taxa C and D. And then, of course, this node here will be the ancestor, the inferred ancestor of all these taxa. The root is at the base of this tree, and it's rooted because we have included an outgroup. This outgroup can be one or more taxa, and they are distantly related to the in-group. So the in-group is typically the group that you want to study, and the group for which you want to know what the phylogenetic relationships are. So we include an in-group, an out-group, to polarize the in-group. By doing so, we can infer then, or determine rather, what the phylogenetic relationships among these in-group taxa are. So just to say again, so this is the root, and that's the common ancestor to all taxa. Sometimes you see in literature that a program requires a bifurcating tree. It just means all the nodes split into, all the nodes have two branches. When it's multiplicating, it has more than two terminal nodes, so there are more than two species or taxa that shares that internal node. It is also referred to as a polytomy. So you don't know what's the relationship in this group among the different taxa, but you can say that they all have the same ancestor. So terminal nodes are also referred to as leaves, tips, external nodes. The internal nodes are the extinct ancestors. So it's not sequences that are available, but just for interest's sake, one can actually synthesize or simulate the sequence that can represent these taxa. But that depends on your data. A rooted tree is a tree that has an outgroup and then is rooted. But you will also see sometimes unrooted trees. So we also refer to those as network trees. The only thing one can say here is that these two species shares an ancestor, these two species shares an ancestor, but we don't know if these two are phylogenetically distantly related to these three. For that, we need outgroups. So how does one determine, or how does one find the outgroups? So first of all outgroups are distantly related taxa to the in-group. So these outgroup taxa can be taken from literature, or for example, if you want to know what are say the genera in a family, you can use as an outgroup a taxon that's from a different family, but you know it's not so distant. So, for example, you cannot root a tree that has mammals with bacteria. That's not going to work. It's too far away. Or if you want to know something about the evolution of phylogenetic relationships of bacteria, you can't use a virus. It's just too far away. So it should be something that's close and distant. And typically we find in literature from previous work what those outgroup taxa can be. You can also do blast searches. So if you compare your sequence against GenBank, sequences in GenBank, those sequences that have less similarity, you can test those. But we test the outgroups. So I would typically take more than one outgroup, so many outgroup taxa, species, and then see what is the effect on the tree. Sometimes when you use an outgroup that's too close related, you will have attraction from that, or that outgroup will attract sequences in your in-group, especially if there's long branches. Okay, so I will just take a moment here and stop for a moment because there are some questions. Adam?
I think you did sort of answer my question, but I just wanted to because if you're constructing a phylogenetic tree and you're looking at subgroups of a specific species, so I'm working on the Pretoria, for instance, and we have multiple morphotype groups, and you want to distinguish between them and see if the different morphotypes do cluster together. So if you want to use an outgroup, would you not recommend that I would use a species within the same genus of the Pretoria? Would that be too closely related? Would I rather use something that's in the same family?
Yeah. Okay, so the question is about, in this example that Adam just mentioned, he wants to know about the relationship of species within the genus, and he asks if he can use a species within the genus. That really depends on the phylogenetic distance of that species that you want to use as the outgroup. If you know that that species is very distant to the species that you are interested in, then you might be able to use it. I've used it in the past because we were interested in an in-group that is phylogenetically very distant to my outgroup. I could use an outgroup that's within the same genus to root a tree. And for those of you that's online, sorry, I'm looking at that direction because some people are sitting on that side. But if you want to know the phylogenetic relationships of all the species in a specific genus, then you would rather use a species that's not in the same genus, outside of that genus, but not too distant. Because if you put something there that's going to be that has very low sequence similarity, it's going to cause noise in your data set. And it might be that your input just collapse. So there's no solution. And we'll see that in your bootstrap values, or if it's a Bayesian analysis, in your posterior probability values. Is there any other question?
The same question was answered in the chat also by Ben, so we could go ahead.
Great. Good. So let's move on. So this is a bit of an old slide. It is more to do with the fact that phylogenetic systematics comes for many years, and there are many different ways that people have talked about cladograms and phylograms. So I have two books here. The one was published by the Systematic Association, and the first author was Kitching. It's a very old books, and this is one that was published by Henneth himself. So they talk about cladograms in those books. Many of the papers that we read, they just simply talk about phylograms. I just want to clarify that it's not a disaster if you use phylograms instead of cladograms, but typically cladograms show the branching order. So we don't know, it doesn't show the distance or the branch lengths. For example, here we have a tree. The branch length between A and B is the same. And if you add the branch lengths from A to the ancestor and the ancestor to C, they will have the same length. But it's not about the length, it's about the order of the taxa. How are they related? Moving forward, we use phylograms to show branch order and branch length. Now you will say to me, but this one looks the same as this one here. And that is because it's an ultrametric tree. So there are two categories, additive trees and ultrametric trees. And I don't want to go into all the detail, but just to say that in this case, the distance from A to the common ancestor of A and B is not the same or equal to B from B, no terminal node here, to this ancestor. So this branch length and this branch length is not the same length. And that has to do with your substitution rate. It can also be due to a time effect, because if you think about molecular clocks, it is assumed, many times wrongly assumed, that if you have a longer branch length for one taxon versus another one, that the one is older. This is actually something you have to test. There are many methods that one can use to test whether this is actually due to age or just substitution rates. So substitution rates will also change the branch length, because if you have one taxon with a high substitution rate versus one with a low substitution rate, in other words, the gene evolution, the rate of mutation in these genes between the two taxa, same gene, that it differs, that will affect your branch length. And we have models to compensate for that.
Martin, before you continue, there's a question, and maybe you can use this to talk about it. They say, when to use the common terminologies. The first one is lineage. Then when will you call it a clade, a group, or an ancestor?
Let's go to a slide where we will talk about it.
Okay, so you're aware of it.
Okay. Then I just want to say, so this one, in this case, the rate of evolution is the same. So the distance from A to the common ancestor in terms of the branch length is the same as B to the common ancestor. So if you had an evolutionary rate that was the same in all three taxa, the distances on the branches will be the same between taxa, the species that shares a common ancestor. So in this case, A2, the common ancestor of A and C, would be the same as C to the common ancestor of B and C. I'm going to skip this tree just for now, because I want to say something about lineage. So a lineage is all the descendants from the common ancestor. So if this is the common ancestor of F, A, E, and D, this will be one lineage to F. This would be a lineage to A, and from there to E would be one lineage. So, for example, humans, Homo sapiens is a lineage, can be seen as a lineage. Chimpanzees can be seen as a lineage. A group can be anything. It can be, this can be a group, F and A. E and D can be a group. F, A, E, D can be a group. So that's just what are the elements in that group. Okay? But we have specific words to describe different groups and their relationships. So that the differences, the relationship between taxa within and between groups. A monophyletic group represents a group that, or species or taxa that shares an ancestor. So if they share a node, in this case A and F shares a node, that will constitute a monophyletic group. But there are different levels. Here we have E and D being a monophyletic group because it shares a common ancestor at this level. But the same can be applied to F, A, E, D. They share a common ancestor at this node. Therefore, together they can form a monophyletic group. So it's important to refer to make it clear what you are referring to, because at this node, all of these taxa are monophyletic. They belong to a monophyletic group. They can also be seen as a clade. So Fa can be a clade, Ed can be a clade. So if you have a monophyletic group is a clade. So we don't talk about a monophyletic clade because that is per definition, clade is per definition a monophyletic group. So right at the root here, all of this can be seen as monophyletic because they all share a common ancestor. F and G is not monophyletic because F and G doesn't share a recent ancestor like F and A. They do share a common ancestor here. So I'm talking about G and, let's say, A and G. They do share a common ancestor here, but that would be then including all of these taxa. So one cannot say G and A is a monophyletic group. One can, however, say that G and A belongs to the monophyletic group that also include all these taxa. One can, however, say that A and B is paraphyletic to each other. So G and A, as in my previous example, is paraphyletic to each other. So it's very simple. So things that are not in a monophyletic group are paraphyletic to each other. If you want to use those terminologies. So I hope I have answered the question. So clade, lineage, monophyletic group. Monophyletic group is a clade. In terms of paraphily, if you do not share a common ancestor, if there's any taxa that should be excluded from this group, then it's paraphyletic. So what I mean is, if we say A and B, that cannot be monophyletic. It's a paraphyletic group because you have to exclude E, D, G, C, and F in this case. Okay? Therefore, their relationship is paraphyletic.
The issue of ancestor, that none of these could be an ancestor, but it's only the internal node.
So the internal node is the ancestor, but it's the ancestor of F and A at this node. At this node, this is the hypothetical or inferred ancestor of F, A, E, and D. And as you go down to your backbone, you will have more inclusive ancestors. So at this point here, this is the hypothetical ancestor or inferred ancestor of all of these taxa here. So here, it will be the ancestor based on this phylogenetic tree of F, A, E, D, G, C, and C, B, and H. Cool. Is that okay? Let's finish this. This is also relevant to trees. So when we have a node like this one, where the branches are not bifurcating, but multiplicating. So we have a multiplication here. So we typically say the branches collapse into this node. That node is then a polytope because we have many taxa that shares the same ancestor. And there's no way that we can say what is the phylogenetic relationship between F and A, or F and E, or F and D. The only thing that we can say is that they share an ancestor here, so they form a monophyletic group here with G, C, and B. But it's not like here where we have a bifurcation where we can see that G and C are more closely related to each other than they are, B is to G or B to C. Here we cannot say anything about the phylogenetic relationship. We can, however, see that they share an ancestor. Therefore, we can only say, and that's the only thing that we can infer from this tree, is that all of them has shared at some point in evolution, in some point of its evolution trajectory, it has shared an ancestor that is common to all of these taxa. And this becomes very relevant to the interpretation of bootstrap values. I can't see the questions if someone can read it.
No, it's fine. No. Go ahead.
Oh, can I speak? Okay. Just going to fix something here.
It's fine. I will tell you this question.
Okay. Okay. Good. Then there's also some confusion as to what a basal group is. I don't like to use this word or this term, but just to clarify it. So here, the basal group is typically outside of the different groups here. So let me just rephrase this. So in this example, we can say that H is basal to F and A, or D and E, or G and C. So it's distant related, doesn't share a direct ancestor, so a recent ancestor. One cannot say that B is basal to H, because B doesn't share an ancestor, a recent ancestor, with H. So it is taken then that H, if it's a basal taxon to F and A, that it is an older lineage. So that's all that this means. I will come back to this when I talk about relationships. Then another terminology that we use, and this is really make it much easier. You can say that F and A is a group, and this group is closely related to E and D is a group. Say F and A is a monophyletic group, and E and D is a monophyletic group, and they are closely related to each other. Or you can just simply say F and A is a monophyletic group, E and D is a monophyletic group, and they are sister to each other. Now G and C here, they are monophyletic, they comprise a monophyletic group, represent a monophyletic group, but they are not sister to FA or a monophyletic group that includes E and D. In this case, one can only say the only time that this is relevant in terms of sister groups is when we refer to this whole monophyletic group that includes taxa F, A, E, and D. And if that group shares a node with the monophyletic group that includes taxa G, C, and B, we can say that this whole group is a sister group to this whole group here. Okay. So it makes it just quicker to write or to explain. Most people will understand if you say, if you've defined what is your monophyletic groups, and you didn't say they are sister, we know they form a sister group, so they share a node or an ancestor, the most recent ancestor of both these groups.
Martin, there has been a number of questions up to this point, and I think it would be helpful if we can deal with some of them. The one question was, are multifurcating trees more or less common, more common than bifurcating trees? When would we see polytomies in nature? And please give an example. And Rob has already said, well, it's usually because it's poorly resolved and we collapse the nodes. It's because we don't know.
Yeah, and thank you, Rob, for that. And also thank the group members can also give answers. As I said, it's interactive. I'm just leading it. So thank you for those that are also answering some of the questions. So the polytomy here, that typically is when you do not have, if your data is not well structured, or if your data doesn't have strong phylogenetic signal. If you here, for example, take sequences that has very low or very high similarity rather, you typically can't get a resolved phylogenetic tree. So you can have a mixture of sequences where some taxa, it's just the same, but different, but not different enough to make them a monophyletic group. So it's good. It's similar enough. It will, if there was no similarity, no variation, they will group together. But there's some variation that can set them apart to this group here, but not to resolve the phylogeny here. Then you get a polytomy. So taxon sampling also important. If you do not have, if your taxon sampling is poor, you will get this situation where you have multiplication. So it has a lot to do with your sequences, and then of course, what taxa are you including? This we see sometimes. I mean, in my own research, I had trees where, because the gene that you use doesn't provide enough resolution, so there's not enough variation here, or it might be even that you have too much, but let's just keep that on the side now. Then you can't separate these taxa, they are the same. And I had this in some of my papers. If you add another gene, it might resolve this polytomy here, or if you include more taxa, so sequence genes or taxa.
So if the resolving power isn't good enough to resolve that polytomy, would it then be wrong to say that if, for example, is the sister to the BC modified, you can't call them, can you call them sister groups or one group?
So you cannot say this group, so FAED, you cannot say it's a sister group to GCB, because for that you need monophyletic.
Okay, there was another question related to this example where it says, you know, if C is your, B is your unknown, how can you say with the polytomy, can you still deduct something about the relationship between B, C, and G?
Oh yeah, no, definitely. You can say that B is basal to G. You can say that GC is a monophyletic group and it's a sister group to the taxon B. So we know that B is related to GC. Here we don't know what's the relationship. So in this case, if it was speciation, one would think that, okay, B, so there's this one species, then we had over time, due to whatever speciation mechanism, whether it's allopatric speciation or sympatric speciation, that the ancestor of GC was separated from B, and that they have then evolved into two different species, that they share the same ancestor here.
Okay, thank you.
If my answers are not clear, please tell us. Good. I'll come back to that. So another thing that one should remember when you interpret trees, sometimes you have two trees that you want to compare. And then just based on topology. So topology means just the structure of your tree. It looks like they are different. But it's just because of the way that it's presented, or the fact that it was not rooted, or the fact that branches were just rotated, not swapped, rotated. So here I have two trees, and we, per definition, two trees are different from one if one tree, specifies at least one partition that is not present in the other tree. Now I'm going to ask you, do you think that these two trees are similar in topology, or the same, or do they differ? And please note, these two trees are not rooted. So I don't know if we can have a poll, but perhaps just in the chat, I don't know what would be the easiest. Just a few of you can say, well, they are the same or not. Some actually. So my question to the audience, or to the class, the group, seeing that it's not a lecture but a discussion, the discussion members, are these two trees?
Well, there's one that says these two are like day and night.
Day and night, okay? These two are night. Thank you, Marcel.
Yeah.
Christoph says same. Ben, I think they are the same. Someone here, I think they're different.
I have a question before I ask that. So it says note these two trees are not rooted, but to me it looks like the second tree has an outgroup C and therefore it's rooted. Is that not true? Did I misunderstand that?
Yeah, it's not rooted. I didn't put a root. And neither did I root a tree in the program.
Oh, so when you add an outgroup, it's not necessarily rooting it.
Well, if you add an outgroup, you want to root. But when it shows like an outgroup, it doesn't mean it was an outgroup.
Yeah.
And that you will
I will
root.
show you with the program how to root. But typically your outgroup, because it's distant to your in-group, it will go to the basal side. Because it's
So
distant.
then,
Let us just okay. Good. Okay, we will stop this now. That's always a fun exercise. In this case, these two trees are actually the same. And the reason is because all the partitions, so all the nodes share the same taxa. So for example, here we have A and B that shares a node. Here we have A and B that shares a node. Here we have ABC that shares a node. ABC, it shares a node. So you have to actually, I was a bit naughty in the sense that I didn't tell you to just open the tree, because it's a network. Okay. So that in fact, these two trees are exactly the same. If you look at how they share ancestors or nodes, you will see that it's not like A and I shares a node there. In all of them, if you open it, if you just make a network of this, you will see that they all both share the same nodes if you compare it to each other. So what I mean is, DNA shares the same node there, then DNA there. So DNA is monophyletic. Well, let's stop, not monophyletic, but they have the same grouping. Yeah, that's the word, grouping. And here we have ABC, as I said. So ABC, if you draw a line like this, F and G does here, and FG is the node that is here, and we have I and J together, I and J, and sorry, I, J, and H together, I, J, and H together here. Then together they form a group F, J.
So somebody is
J.
asking, sorry, can we guess the ancestral node?
No,
No.
you can't guess it. Why would you? No, you can't. You cannot guess it. Okay, so be careful how you interpret trees and compare trees. We will get back to this. Okay.
I was asked about the difference between rooting and outgrouping. So is rooting then the act of telling the computer to keep this to the side and then move this to the
So we talk a bit loose about generically about rooting. So the root is this point. Okay, so that node is the root of the whole tree. This outgroup is used to root a tree. So rooting a tree can be a physical thing that you do using the software. So you say on this branch, root my phylogenetic tree. I will show you in a fig tree how this is done. Okay. Perhaps we can move forward to branch swapping and rotation. So there's a huge difference between branch swapping and node rotation. It is okay to rotate a node, not a node, sorry, a node, that should be a D, a node, but it is not okay to move a branch. So if you do a phylogenetic analysis and you think, oh no, but F is actually, I think F should be with B. You can't take that branch and stick it there. You can do it with programs. It is doable, but it's not correct. You cannot, then you alter the tree with the topology. By altering that, you're also altering the phylogenetic relationship based on your data. So you are actually misleading, well, not only misleading, you don't do that. That's my point. But you can rotate around the node, because if you rotate the node, so you just rotate it so that E and D is rotated, so D will be above E. You have not changed the phylogenetic relationship here. You've only changed the how this tree looks like. So not a phylogenetic relationship, not a topology in terms of the relationships. So that you can do. You can do this anywhere you like to, on any node. You're not changing the phylogeny. You are just changing the balance of your tree. So here I have, what did I do here? A, B, and E. Here I've done what I told you not to do, just as an example. I have to apologize. This is not entirely correct. So what I explained here. So here you can see A is now part of the group DE. But here in my original tree, so that's my tree from my data, A forms a group with F. So I've moved the branch. That is something you may not do. The same goes for F and B. G and C, there's no change. So again, my point is just, you're not allowed to move branches because it doesn't fit your hypothesis, but you can rotate your nodes. The reason why you would rotate your nodes is because sometimes you have a tree, say from one gene and a tree from another gene, and you want to compare how these gene phylogenies compare with each other. So you can rotate a node so it matches the order of the one tree. Or if you have a phylogeny, you conduct a study where you want to compare the phylogeny of a host species or host species with the phylogeny of parasites or pathogens. You can rotate the one tree, the nodes, so that you can actually compare the two trees in a way that makes sense to you. Okay. So someone is drawing on my screen. Please do not do that. So you can, I think you get the point. Any questions about it? Okay, so I'll move forward. Okay, this is just, I cannot, this is okay, we've dealt with this. Okay, another thing that people sometimes misinterpret is when you look at the relationship, sometimes because this wolf here in this phylogeny that includes human, wolf, alligators, and fish, just because this wolf is here at the bottom doesn't mean it is more closely related to alligators than it is to humans, or that alligators are more closely related to wolves than they are to fish. Okay. So in this case, human and wolves, they share the most common or most recent ancestor. Alligators and humans and wolves share the most closely related ancestor, a common ancestor. Therefore, humans and wolves are more closely related to alligators than they are to fish. So here we have the branch, the node just rotated. Some people think then, in this case, humans are more closely related to fish just because it's there. But you have to see it in terms of the ancestry. They are more closely related to alligators or to wolves than they are to fish. Okay, so we will now move to the demonstration. So these are just the clarification of terms and explanation of interpretation of trees. So if there are questions that you want us to deal with now, we can do that. And then we will go to examples and demonstration. Yes?
Can you go back to the trees that were they are they different that they were saying? I think I may have misunderstood. I have a misconception, and I think it's been exposed now. So it seems to me in the second tree that the speciation event between the common ancestor of A and B and so it seems to me that that happened earlier than in the first tree.
No.
No.
It's not the same.
Sorry?
The distances are exactly the same.
So to the people online, the comment here from one of the students is that
Yeah.
what did you say?
So it seems to me that in the first tree, we have whatever the common ancestor for everything is. And we see that speciation event of the common ancestor between C and A and B. And that happens differently in the second tree from how I'm reading the trees.
No, so just open a tree, but I think if
We will
We can
explain to you later.
I think we
But
can.
it is, you just need to open the tree and not think of it as, it looks like rooted, but it's not. And you need to keep that in mind.
So here's another example, very similar to, but in this case, this is actually rooted. But it's just because we have not, you can ask the program to balance your tree or to ladderize it. Then it will take this like, here it is actually balanced, here it's not. So it will just make it like a step ladder. And then you will see most trees look the same. And if you rotate nodes, you can get these trees. They are exactly the same, these two trees.
There's just this one question, and we need to deal with it, is when will you be collapsing nodes? How do you determine whether a node should be collapsed?
So to answer that, I'm going to show you this phylogenetic tree. This phylogenetic tree is actually something that I use in for demonstrations. So it's actually part of a much larger phylogeny dealing with the phylogenetic mutations. So you do not collapse a tree that's generated from your data. So when we present our phylogenetic trees, we typically do not collapse the branch into a polytomy unless you very clearly state that you've done that because to, one, illustrate something about, you don't care about the taxa where you have that polytomy, where you have actually induced that polytomy, or if you do not have support, phylogenetic, statistical support, either by way of bootstrap or posterior probability values or whatever the index you use. If you do not have support for that, you can collapse it. But then you say that. So here's to further illustrate what I mean is here we have a phylogenetic tree. There are very different taxa included in this tree. They belong to whales, porpoises, and dolphins. The dolphins forms a lovely monophyletic group, strongly supported by 99 bootstrap value. The porpoises as a group forms a monophyletic group with a 92 bootstrap value. So I've done bootstrap on this. And this is a maximum likelihood phylogenetic tree. And the whales forms a monophyletic group that's supported by 82%. Now, if we focus on the dolphins, you will see very small values, values lower than 70%. There's 49. I included them on purpose. So there's a 40% bootstrap value, which means in your sampling, and I don't want to go into the detail of bootstrap. We will deal with that in a later session. But in your sampling of your bootstrap sampling, only 40 trees or 400, if it's out of 1,000, had this relationship between these two taxa. So clearly then, there's no support. In this case, only 27, if it was 100 bootstrap reps, only 27 trees, it was 1,270 something trees had this relationship. So no support here. Okay. Now, you can leave this tree as is, and that is what I would do. Or if you want to make it easier and clearer for your reader, you can collapse these nodes into this node. This node. This is 60. You will not collapse this node because it's 100%. So that is just to make it visually easier to interpret, especially for people that do not understand a reader that's not a phylogeneticist or doesn't deal with this on a regular basis, then they can see that there's no phylogenetic relationship. But often we do include it just to say that this is how the phylogeny, this is the resulting phylogeny from our method that we used based on our data. But then you say in your legend, your figure legend, that all nodes that received a bootstrap value of less than 70%, all those nodes were not supported, or you can say that bootstrap values less than 70% are not shown on a tree. But in your mind, and that's what I tell our students, what you must see is no phylogeny here, or no relationship, but a branch that collapses into a node that is highly supported.
On the same topic, there's a question that says, is it okay to collapse a node with multiple sequences of the same species?
Oh, yeah.
Because if there's no support, there's no support.
Well, sometimes we want to know, just a scenario, I go to Sumatra, I collect fungal species, but many isolates, because I know this fungus is in that forest, and I sample like a crazy person, and I sequence 200 of these samples, and they're all the same. But I want to show, I want to say that I have sequenced all of them, so the major fungus that occurs in that region, if it's a pathogen, the major pathogen or the pathogen or fungus that's prevalent or most frequently found is this species. Then I come in all our sequences on a tree and just collapse it so it forms a triangle, and then say this represents 300 isolates or sequences from 300 isolates. So that is fine. By the way, you will also make available your raw data or your raw alignments on whatever repository you use. So anyone can go back to your sequence alignments and then see, well, okay, you've done this, this is why you collapsed it. So you can collapse it into a single branch, but if you want to show off, you can collapse it into a triangle, so you will have a big triangle if you have many sequences.
Okay, then this is a bit unrelated, but Marcel is asking, it might be a little nitpicky, but wouldn't it be more accurate to say ancestral population as opposed to most recent ancestor? Most recent ancestor implies a single individual is responsible for an entire lineage, which does not feel right.
Right. Marcel, you are actually correct. It's just the way we talk, but of course that was the ancestral population of that species. So evolution doesn't happen in one individual, it's in the population, and it's through that process that you get speciation. So you are correct. It is a bit nitpicking, but that's fine. But what is important, what you show here is one should not think as an ancestor as it's not an individual that was the ancestor of crocodiles. It was the population. So you're right. But it's shown as one node. So therefore we refer to it as the ancestor. Okay. Yeah, good. Right. Perhaps we can move on. So just, I want to ask the people that's here in the room that's helping me, will we sabotage?
Someone just on, I suspect, would yourself.
Okay.
That's the usual.
I'm starting to think it might have been me. Okay, good. Right, okay, so let's get back to our example. For those of you that have a little bit more time, we can do that. So again, this is based on this paper. It's a very old paper, 1998. I've looked for more recent papers, and there are, but we are now moving into genomic data, so phylogenomics, and so on. But I wanted to get a tree where I can actually show you bootstraps on the tree. And just because we also had a question about bootstrap values and so on. So this is then a phylogeny. Doesn't matter whether it's from morphological data or sequence data. These values here are bootstrap values. So in this case, that 55% here means this whole group collapsed into this node. There's 57, so this group collapsed into that node there. This group collapsed into that node. So you have three groups. It forms a polytomy here. We do not know if this group is the sister group to this group. We don't know if this group is basal to this group or a sister group to this whole group. So because your polytomy will be on that node. So the same here. We don't know what is the phylogenetic relationship between these species. We know that this species is a mono, these two species are monophyletic, and this branch here will collapse into this node. This branch here will collapse into this node, and then all of them will collapse into there. So you can just as well say that this branch join that node, this branch will join that node, and all of these branches will join that node, but it will be just, it's like a star. There's no resolution. So nothing can be said about the phylogenetic relationships of the dolphins in here, except that Torsiops truncatus and Delphinus dolphins are in the same monophyletic group. So they share a common ancestor or the population for that.
Okay, so there's a question, what is the threshold needed to collapse a group? And I think that's a very debatable thing.
It is very debatable. But so if we just look at literature and the studies that were done, simulations, we would take anything that's lower than 70% as no support. So most people would say, well, any bootstrap value lower than 70, we don't have any confidence in it. I personally do the same. And that's based on these papers. It's not just a thumbs up value. Then you can have categories, and you say, and one can see, we can state that 70 to 90 is good support. The node is likely reliable, and then 90% and up, you have high reliability, so strong support. And one can code your branches in different colors. You can add different color dots on your node to indicate the percentage ranges. The point is, I collapse in my mind, when we talk about phylogenies, anything lower than 70% based on these studies, so that there's no support. So you also have to interpret it in the context of your phylogeny, in the context of, there might be support, external support for that group, although the bootstrap doesn't support it, because remember, your bootstrap is just a value that's generated from your data through a process of sampling and then making consensus trees for trees based on a sampling strategy.
Okay, I think we've been busy for an hour, so I would like us to start wrapping up a bit. But it says, when doing phylogenetic analysis of the same species, does one construct the tree based on a single gene, or can one construct the tree using complete genomes of the isolates? And it's always better to have more data.
It's always better to have more data, but it's about what your question is. So if I realize there's a whole diverse group of people here. But I can tell you in fungal taxonomy, for example, we use a genealogical concordance approach, and we're moving towards that in bacteriology. So you not only have multiple samples or isolates in your analysis, but you also have many different genes in your analysis, and you're actually comparing your gene trees with each other to separate taxa based on genealogical coordinates. So groups that form groups among all these trees. I think we are moving away from a single gene, because we know a phylogeny based on a single gene doesn't necessarily represent this phylogeny of the species, but the phylogeny of the gene. And by chance, that phylogeny might reflect the speciation or the evolutionary trajectory of the species. It doesn't have to. So the more genes you include, the better.
Just to start wrapping up, Martin, you've seen the questions that was posted in the Google Sheet.
Yes, I did.
Are there any specific ones that you feel you want to highlight? I know there were a lot about bootstrap and how does bootstrap work and things, and my feeling was that that's probably a whole session on its own.
Yeah, and I want to thank those of you that send us some questions, and after the session, please send more. We will have more sessions, I hope. I don't want to I don't want to focus on one specific question, because many of them I think I've answered through this talk. Some of these questions were not related to our reading of trees, and then we will deal with that at a later point. But I've tried to cover those that are related to the specific session. I want to also say that there were some interesting questions that we have not thought about actually, and that needs really actually a session on its own, like constructing phylogenetic trees using functional diversity traits. So in terms of ecology, you can apply phylogenetics in a diverse way, or in diverse fields. So we can look at that as well. I also want to ask that if you ask a question, you need to give us some context, because just to ask, can I use the IT sequence in my chapter? I don't know what you're talking about. Fungi? Is it about the gene? So a little bit of description is needed to really interpret the questions correctly. What I have suggested to my colleagues here and the fantastic SASSB group of students is that we perhaps need a session where we start from the basics, because we had questions about sequence quality and trimming of sequence. So sequence alignments, do I trim my sequence? How do I concatenate sequences? Those are very important things, because if you do have bad data, have bad sequence alignments, you will have bad phylogenetic trees. So that, far as for now, I think that's all I can say. Oh yes, one thing I want to highlight is, yes, you can, one of the questions, can a phylogenetic tree be generated for genus level identification? The answer is yes. I have two examples here. One that was done by a group looking at fresh water mussels. So they've discovered a new genus, Nancungania. Gen-Nov means null genus or new genus, which they then describe as a new genus, this group here, with two species. And then from our own group, we use phylogenetic analysis but at a genomic level, so phylogenetic approach, to first identify a new genus. So this genus is called Mixta. It was previously thought to be part of a bigger group, but this whole group here that I'm highlighting here was separated in three groups based on monophily, and then we discovered, well, we identified a unique genus that was described as Mixta gen. So yes, you can.
I think that brings us to the end of this session. But before we leave, we just want to give Abby a moment to speak about the student group that we have for SASSB and how all of you that are here and interested can actually join in the fun. So Abby, over to you. Abby? Sorry, I can't unmute myself, sorry. We will help you with that. There you are.
Hello, I think that's work. Can everyone hear me?
Yes.
Excellent. I won't take long, but I just wanted to quickly jump on. I did post a link in the chat, and I'm going to post it again now. Thank you very much, Martin, for this talk. It was incredibly helpful, and I hope everyone has found it useful. In general, we basically, what I just wanted to say is we've started a WhatsApp group. It was originally started mainly for kind of students looking for help, but we're kind of treating it now also as just a general place for people who are getting into systematics, who are getting into phylogenetics, anything like that. And want a place where they can talk to people, ask for help, commiserate about things they're struggling with, anything like that. I've just sent a link to it in the chat, and basically just feel free to join. The idea of it is very much just to have a place where everyone can talk. On top of that, we will make sure that we advertise any future seminars like this. So the general SASB seminars, any more like meetings like this or training sessions like this, we will be advertising on there. And on top of that, us as the students are also organizing, we're trying for once to twice a month to have like an informal meeting where students get together and present on their work, which you are also always welcome to come and join to. We're also, next year is going to be another SASB conference, and kind of in the lead up to that, we'll make sure that we post all of the information on there as well. So it's a really good place just to keep in the loop about anything like these. So please feel free to join, and hopefully we see you soon.
Yes, thank you very much. And on this WhatsApp group, we definitely want to have some feedback on whether you found this useful, because we need to decide whether this is something that you can continue with. I am very pleased with the attendance of today. So we really think it's a great thing that students are coming in. But we also want to make sure that it is something that is of interest to you and that you can learn from, because we can just go on and nobody would be interested, and that would be a bad thing. So there will be a link for feedback and suggestions for future sessions in the next day or two. So be on the lookout for that.
Yeah, thank you, Forrest, and all of you that has worked hard to do this. The one thing is we didn't get to the demonstration. I realized that. Perhaps I talked too much. But we can perhaps in the next session focus less on this presentations and more a little bit more on the demonstration. But I think we needed to set the foundation and see how we can go from here.
And as we said before, this session was recorded, so we will post it on the SASSB YouTube channel. So soon you would be able to go back to that and have a look, or tell your friends to go and visit it and have a look, and also use it for revision before we do our next session. So, but there's also some very interesting talks on there, webinars that we've had with experts from all over the world. So you're welcome to have a look. And with that, thank you to everybody and have a good evening.
Thank you to you all.
And thank to Martin for doing this for us.
It was fun. Thank you, Forrest.